Great, thanks very much for the introduction. and really thanks for the opportunity to come and be a part of this important meeting. I think actually the patients and patient advocates are my most important audience, because you're the people that we're serving effectively. So my title, a bit like Uta's, was quite a broad one. And so what I'm not going to do is give you a complete overview of biomarkers in bone sarcomas. But what I'm hoping to do is to explain to you what we really mean by biomarkers and what I think are the opportunities for the sort of molecular studies that we're doing and how we really need to advance these more quickly than we are doing currently for patients. And I move forward by doing that. Great. So I guess the first thing is really, what do we mean by a biomarker? So a biomarker is actually anything that you can measure that's an indicator of a biological process. So a biomarker is a healthy biomarker, or it can be a disease biomarker. So I think we've probably all been to the doctors and had our blood pressure measured, our weight measured, or our pulse measured. And these are biomarkers of our health state. So this is what we mean by a biomarker. And biomarkers come in lots of different flavors, so lots of different types of biomarkers. And already in the clinics, we use biomarkers to help us diagnose patients and in some cases stratify patients for treatment. But in bone sarcomas, biomarkers are used to, particularly I think at the moment, improve diagnosis or help people to, clinicians to diagnose bone sarcomas. So physiologically, there are symptoms that patients present with that are biomarkers of their disease, of their cancer. And on the Bone Cancer BCRT website, there's a nice figure there that summarizes these different sort of physiological symptoms. And these are biomarkers. These are indicators of potentially bone cancer. And histologically, a pathologist will look at a piece of tissue, look at a tumor down a microscope and see the pattern of cells. This is a H&E stain, so these are two different kinds of bone sarcomas stained with hematoxylin and eosin. Basically, there's sections through the tumor showing the actual different patterns of the cells in the tumor, and this is this is used by the pathologist to help define the differential diagnosis. We've already heard about the sort of imaging and how we use imaging to diagnose this is a normal bone scan, and you can see these look quite different, and this helps with the diagnosis of bone sarcoma. So these are biomarkers, and biomarkers that I think hold a lot of promise for future outcomes for patients, to improve outcomes for patients, are molecular biomarkers, and I'm going to try and explain a little bit more what I mean by that, and where I think there's some good evidence that these might be used in the future to help patients. So molecular omics markers are really what I'm going to be thinking about and talking about. So just to try and put this into context a little bit, not to go into too much detail in this diagram, but molecular omics in cancer, we mean pretty much any kind of process that is happening within the cell of the cancer that affects how that cell then behaves. So within most cells in the human body, there's a nucleus, and within that nucleus there's DNA. And this DNA is packaged in chromosomes, which basically this is the information in the cell that tells the cell what it's going to do, what its behavior is going to be. And as Uta's already said, in some sarcomas, we know very specifically that the DNA abnormality that drives the initiation of the cancer. So in theory, that should be exploitable to actually develop new therapies, but that's not always as easy as it sounds. But for many of the bone sarcomas, particularly Ewing sarcoma, which is a sarcoma that I work on in the lab in Leeds, there are actually very few genetic abnormalities, and much of the regulation of the diversity that you see in patients is actually at the what we call post-transcriptional level, so the regulation of the cell after the DNA, so the production of RNA and protein. And these processes are called epigenetic processes, And increasingly in the science community is understanding the importance of these epigenetic events in driving a number of different diseases, including cancer and bone sarcomas. So in terms of the omics, what we're really meaning in terms of studying DNA, we talk about genomics. So this is studying the DNA of a cell. If we think about RNA, which is the message that goes from the DNA to the protein. The proteins are the bodies that do the job in the cell, but there needs to be instruction for the cell, the proteins in the cell to behave in a certain way, and this is regulated through the RNA, and we talk about transcriptomics. And then actually proteomics, so how the proteins actually work. And there's lots of activity across the world studying the proteins and the transcriptomics, the proteins and the RNA in cells, including bone sarcomas. There are lots of different omics arising, so you'll hear about metabolomics. And just to clarify, omics really means the sum of anything that's happening inside a cell to make that cell behave the way it does. And because we know cancer cells don't just act in sort of isolation, they interact with each other and interact with cells of the microenvironment. Omics could include anything that's about the tumour cell, but also the microenvironment cells as well. So there's lots of excitement about different omic approaches. But in a nutshell, I think where we've got opportunities is to understand the heterogeneity of the bone sarcomas themselves, to understand the tumour cells that are actually making up the tumour, but also the microenvironment cells that influence the behaviour of the tumours. By understanding these cells, their tumour and microenvironment, we can identify potentially different cells of different behaviours, so cells that are resistant to current treatments, or cells that will respond to current treatments, cells that are responsible for driving progression and relapse, compared to cells that are involved in initiation. And by understanding the roles of these different cells in patients, use their behaviours to identify patients that have low risk, so they may have different treatments to patients that have high risk. And by low and high risk, I mean patients that will do relatively well with current treatments compared to patients that aren't doing well with current treatments. So the goal of these sort of studies that we're doing in omics is to identify sort of more effective treatments to improve outcomes for patients, but also kinder treatments, because patients treated with different therapies, radiotherapy or different chemotherapies, can have side effects. And so we want to have the most effective treatment, but minimizing the side effects as well, so that the patient actually gets the best treatment based on the biology and their needs, but also doesn't accumulate treatment-related side effects, which are going to affect their quality of life. So this is kind of the overall goal of our studies worldwide, I think. So patients and families ask a lot of questions, and this is a list of just some questions that patients and families asked. I'm grateful to Bone Cancer Research Trust and the ECMC, both in the UK, for doing some work on this, just to identify the kind of questions that patients and families ask. And a big one, of course, is, do I have cancer? Is that right? And can I be cured? But then once a diagnosis is made, is this treatment working? Is my cancer coming back if the patient's on follow-up? And these are the sorts of questions that doctors ask when they're treating patients, but they use slightly different words. And also the questions that can be currently in some situations answered by studies of omics, but I think there's opportunities to answer more questions by studying these molecular profiles and omics. So in the sort of concept of diagnosis, we actually already use some mutations and some diagnostic tools, some proteins, to accurately define whether a patient has a specific cancer type. And so these are used to separate patients into different patient groups, and I'll give some examples of that in a few slides. Occasionally we use these to stratify patients for different treatments, depending on how they might do. Actually, there's a lot of evidence on some different biomarkers, different omics markers in the literature that suggest we might use these in the clinic, but they're actually not currently used in the clinic. So I think this is something we might discuss at the end, how we can accelerate some of the scientific findings into the actual clinic. But also in terms of finding targeted treatments and actually monitoring patient response, that's really not routinely used in the clinic at the moment for bone sarcoma patients. There's lots of preclinical evidence to suggest that this could be used, and I'd be interested to see what people think about some of these ideas. So I guess which omic biomarker you should use depends very much on the question that you are asking and on the actual patient. So biomarkers can be tumor biomarkers. So if you have a tumor at diagnosis, you could maybe look at a tumor with different DNA or proteomics or look at the RNA transcriptome to define the patient into specific groups for treatment. Or you might use liquid biopsies. Most frequently, when we talk about liquid biopsies, we mean blood samples, because they're easy to collect. And I think there's lots of promise for using liquid biopsies in the context of managing patients and maybe providing more adaptive treatment, depending on how patients are responding in real time, rather than patients entering into clinical trials for 5, 6, 7 years and following a fixed protocol. And we can discuss how some of that might work maybe later on. So here's an example of a DNA abnormality that has really transformed diagnosis of bone sarcoma, so Ewing sarcoma. This is what we call a gene translocation. So this is effectively, this is some cartoons of the chromosomes that I introduced you to earlier on. So all our cells, most of our cells at least, have chromosomes and that control the RNA, well, that are giving the messages to the cell to how it behaves. And usually we have 23 sets of chromosomes, XY chromosome, and then 22 other pairs of chromosomes that are imaginatively named 1 to 22. What you're looking at here is what we call a chromosome spread from a cell, and this is basically all the chromatin, paired up into the different partners, 1 to 22. And if you look carefully, you can see there's some light and dark banding patterns here. These banding patterns are actually unique to individual chromosomes, and very skilled people in cytogenetics department use these to pair these chromosomes up. And then they look at some of the pairs and see, in some cases, some of the chromosomes look slightly different. And this is how the EWS fusions were first identified. So the fact that there's this really small change in length of these two chromosome 11s here, I'd really struggle to see that. But as a cytogeneticist skilled in this, you'd spot this. And this is the partner 22. This is actually shorter than this one. This simple change is actually what drives the initiation of a Ewing sarcoma in a patient. And it's effectively because you have a break point in these two chromosomes, and you get exchange of the genetic material between these two chromosomes, so that you get effectively a new gene, which has a bit of chromosome 11 and a bit of chromosome 22. And this new gene now will change the expression, change the transcription of the proteins in that cell. So it becomes an aberrant transcription factor. So this is driving the Ewing sarcoma initiation. But importantly, in the clinic, this is very powerful to help improve diagnosis for Ewing sarcoma. So this was originally described by Olivier Delattre And here's a table that just summarizes the different kind of EWS gene rearrangements you can see. You don't need to understand the details of this at all. It's just to demonstrate that these changes in the DNA now, because of these changes in the DNA, we're now able to accurately diagnose Ewing's sarcoma. Prior to this, different tumours that had these same genetic abnormalities were called different things. They were Ewing's sarcoma, named after Sir James Ewing, who described the first Ewing's sarcoma in the leg, or peripheral primitive neurolectoderm tumour, or Askin tumour, which is in the chest wall usually. But we now know these tumours all share this one genetic abnormality, and so their origin is the same, and so these are now grouped together and treated in trials the same. So this is a good example of an omics success, improving the diagnosis of some bone sarcomas. A lot of different sarcomas, not just bone sarcomas, do also have specific fusions that can be used to specifically describe and diagnose the sarcoma type, but not, unfortunately, all. So for a diagnosis, these fusions are very important for some sarcoma types, including Ewing's sarcoma. But for predicting outcomes, so for deciding how patients are going to behave, unfortunately, these fusions don't always tell us very much at all. So in Ewing's sarcoma, although there are a number of different rearrangements, slightly different, the EWSR1 gene, which is on 22q12, is always the same break point, but that partners with a number of different genes in the genome. But it doesn't really matter which of these you have. If you're a patient with Ewing's, you have Ewing's, but these don't tell us whether you're going to do badly or not. So it's not very helpful in the clinic to predict outcome and stratify patients for different treatments. There are some examples of translocations that are important, though. So SICDUX4, actually patients that have this fusion transcript do really badly. And we know this now from several studies in the literature. Even though this is a super rare group of sarcoma patients, these patients always do badly. So actually these patients could get some different treatment maybe to try and improve their outcomes. So most patients with SICDUX4 are soft tissue sarcoma patients, but there are some bone cancer patients who have this fusion, a small group. So, maybe we should be thinking about using this to treat these patients slightly differently to try and improve their outcomes. We can also use biomarkers that are not DNA. I've been talking about DNA biomarkers, but you can also use protein biomarkers and RNA biomarkers. This is just an example of a biomarker that we discovered in Leeds through some studies on tumours and tumour-derived cells. It doesn't really matter what the name of the protein is, it's the idea that I'm trying to get across. So we studied tumours from patients, isolated the cells from Ewing's sarcoma patients that are actually driving the progression and relapse, and then we looked at their RNA profiles and characterized them and identified a panel of proteins, one of which is this neurexin-1. But if you have high expressions of this protein in tumours at diagnosis, which is just shown here, this brown stain is the high expression of this protein. If you have high expression in the tumour at diagnosis, then you do particularly badly. So this is what we call a Kaplan-Meier survival curve. This is survival against time, and these are the patients that have, in this very small study, high levels of the expression of this protein. And you can see these patients are doing much worse than these patients. Now these patients are actually all patients with localized Ewing's sarcoma. So most patients with localized Ewing's sarcoma do quite well. Over 70% will survive to 5 years. But there's a 30% population that do particularly badly. And this biomarker is picking up the population of patients with Ewing sarcoma that have localized disease at diagnosis but do particularly badly. Now, I think there's an opportunity here to try and use biomarkers that are distinguishing this population and to actually treat them differently. Maybe move them into the treatment protocol for high-risk patients. They might benefit from more intensive treatment, but certainly if they're not being cured by the non-intensive treatment, then I think we need to do something. Now, there are a number of different biomarkers now that we know of that can identify this small proportion of localized patients that do badly, the 30%. And there's an international collaboration to compare these. As Uta said, we try to compare biomarkers in different clinical trials. Really a study that's been run through Essen and collecting information, looking at prognostic biomarkers, and a study being run through Birmingham. We should be comparing our data and comparing the biomarkers to find out which biomarkers are most reliable and are going to be most clinically informative in patients. But certainly in the EEC and the Children's Oncology Group, there's been some work to try and summarize the most promising biomarkers at the moment in Ewing sarcoma. And I've only put a couple of references in this presentation because that's, it's not to give you lots of reading to do, but this was one example of a paper that was a group of people came together and discussed all the different potential biomarkers that might be used to improve outcome for Ewing sarcoma patients. So the whole idea behind using biomarkers is really to have a more personalized medicine approach. So rather than taking all patients and giving them all the same treatment, obviously you've got to have the right diagnosis first, that's the most important thing, but then how do you treat patients? Rather than treating them all, actually use biomarkers to define groups based on their risk, whether they're low risk, so they're likely to do well with minimal treatment, or whether they're high risk and need more intensive treatment, or if not more intensive treatment, different novel treatment. The objective is, I think, self-explanatory. It's for more effective treatment, but safer treatments that are less toxic, so we don't have the long-term patient morbidities that we see, with a goal of saving more lives and improving the quality of life of people who actually survive. And using the biomarkers, I believe we can do that based on the biology and the needs of the patient. So if we're talking about monitoring responses to treatment, rather than putting patients into treatment trials and then seeing how they come out at the end, the obvious way to be able to do this most effectively is by using liquid biopsies. So this is attractive because they're minimally invasive. It's easy to take a blood sample. Potentially in the future, we might see tools that patients can use to monitor themselves at home. Certainly that's coming online for some of the adult cancer patients. But I think, it's this minimally invasive way in which you could monitor in real time what's happening to patients, what's happening to you. There's a lot of interest in the scientific community in nucleic acids as circulating biomarkers. The reason for this is because nucleic acids are very stable. Usually we're talking about blood samples, because that's easy to collect. And yeah, it's where most of the research is happening, certainly in bone sarcoma patients. Nucleic acids, this is this sort of DNA or mRNA, the transcriptome that I've been talking about. These are just some of the groups that are involved in these studies in nucleic acids. I seem to have lost my pointer. So this is certainly an area of active research, but I'm not currently aware that this is being used at all in the clinic as yet. But this is because we need to accumulate sufficient information to be confident that the findings in the different studies are meaningful and generally applicable to the whole population. So I think personally that there's an opportunity to use liquid biopsies, to use blood samples, measuring different nucleic acid types, to maybe do earlier diagnosis for patients. Certainly to potentially stratify patients and monitor patients in real time. See if patients are responding to treatment, and if they're not responding to treatment, then maybe adapting the treatment. So I'm just going to give one example of a nucleic acid that's been studied in research setting. This is research that's been done in a clinical trial, taking samples from patients that are treated in a clinical trial, but the samples are taken to do the research. They're not affecting how the patients are treated. I have to say, there are other circulating DNA assays available. This is just an example that I was wanting to illustrate, because there's several groups have studied this similar biomarker and seen similarly promising results. So maybe this is closest to the clinic. So all our cells in our bodies secrete DNA, whether you're a normal cell or a cancer cell. Your cells secrete DNA. And if you are a cancer cell, you secrete different DNA to if you're a normal cell, if you're a healthy cell. So potentially you can use this DNA in blood to see if the patient has got evidence of cancer in their body. And this is exactly what people have been doing. So they've been taking blood samples from patients and detecting tumor-derived DNA in their blood samples. So this particular example builds on this specific fusion transcript that I introduced you to, the EWS fusion transcript in the DNA that's specifically involved in the initiation of Ewing's sarcoma and the driving of Ewing's sarcoma. So to develop the test, you need to have a Ewing's tumor. You need to have the tumor from the patient, to extract the DNA and then identify the specific sequence of the DNA mutation, and then develop a specific assay based using a technique called PCR, which has really revolutionized this kind of assay, but we don't need to go into that. We can if you want. If you want to ask me questions about PCR, we can. But then having designed this assay, using this in patient samples to monitor what's happening to patients. So here we have an example from this work that's been done by a group working with UTA in the EFAT study, where they've collected blood samples. It's easy to collect blood samples throughout the trial design, and then analyze them for these circulating DNA in a patient-specific way. And this is just one example of an outcome plot from this paper that was published by this group, led by Marcus Metzler. And you can see here, this line here, where there's very high levels of this DNA. These patients are doing particularly badly compared to these patients where there's low level of this DNA. So I think this is really an excellent example of how you could use this kind of research to monitor patients and potentially decide, if the patient's not responding very well, maybe we should do something differently rather than just keeping on doing the same thing. So it comes back to this whole idea of a more personalized medicine approach, actually understanding the biology of the tumors and the patient needs and how they respond to the treatment, so that we can stratify patients for better treatment and then improve their treatments in real time. So just to summarize, I think there are opportunities from omics. We already are using it in some way from the diagnosis and prognostic point of view, but really there's lots of preclinical evidence that we might be able to use this for early diagnosis, adaptive treatment, or even personalized medicine. But we have some challenges in doing that. And one of the things is, of course, comparing all the different biomarkers, and we need really to come together to share data and decide which are the informative signatures that we need to use to help patients most effectively. So what we'd call an algorithm of risk, defining all these different biomarkers, not just the omics biomarkers, but also in the context of clinical biomarkers that I sort of mentioned at the beginning. And I think innovative trial design is really going to be key for translating some of these findings to the clinic. We had a bit of discussion this morning about trials, designing trials where we don't have placebo-only arms, and recurs an example of such a trial. I think these kind of innovative trials could maybe progress some of the science into the clinic a bit more bit more quickly. That's really where I was going to I just want to flash that up just to say thank you to all my collaborators and funders. And obviously okay, so if anybody wants to contact me and talk about anything, you can afterwards, or drop me an email or something.
We've got time for a couple of quick questions if anyone's got any questions for Sue.
What do you think, how many years will be available, for example, a urine test that we can check the biomarkers or one biomarker?
Okay. So there is already a Ewing's test to diagnose Ewing's test accurately, the fusion, yeah. So this, I suspect this will be used in your pathology departments, looking for the fusion transcript to diagnose the fusion.
But you meant like for prognostic? Yeah.
Oh, you mean for outcomes? Prognostic.
Like,
Yeah.
monitoring that occurring the disease or not?
Yeah. So I think for curing the disease or not is further away than for monitoring. I think for monitoring, already there's a lot of information on tools that we could potentially use in the clinic. But before you could use them in the clinic, in my opinion, we need to compare those tools and then bring them together and decide what the best combination of tools is. So it's not just the tools, the omics tools, it's also the clinical factors that we already use. But I really do think, I'm looking at Uta because Uta's involved in some of these studies as well. I think there's a lot of information both from the US, from Europe, from the UK, and actually bringing all that information together and having a trialist, a clinical trialist who's brave enough and confident enough in the data to run the trial is what needs to happen. I think the evidence is there to do it. But that's for the prognostication, yeah? But I think particularly the localized patients where you've got 30% who are doing particularly badly, there's several markers that could be used for that already. And I think we need to think how we can accelerate that.
I think I showed in my talk that we are already also using biomarkers that could inform us for drugs that would work. For example, with the GD2 that you could use in these high-risk patients or PRC1. It's a hope, it's a try, because before you haven't done the clinical trial, you don't know whether this biomarker is really reliable, but if you don't try, you can only lose, right?
But I think that's right. And so you also mentioned the marker from Thomas that you're using in your trial, which picks up 20% of Ewing's sarcoma patients. Yeah. No, well, it picks up 20%, Uta, and actually there are other markers that pick up another 10%, and there are other markers that pick up another 15%. So if we bring all these markers together, we might be able to pick up and stratify probably not all of the patients, but certainly a large percent of them. So I think there's a real need for people to come together and bring this data together and then design a prospective trial to evaluate the markers. That's my opinion.
One last question just here.
So changes to biomarker cells, pre-target cells, so to what extent can you correlate that back to genetics or environmental factors which may have given cause to that? Is there any linkage you've established back to those environmental factors?
So there are some studies that have been led by Olivier Delattre, who actually identified the fusion transcript looking for predisposing events that might contribute to development of certainly Ewing's sarcoma. But that's not, those sorts of studies need huge numbers of patients to define underlying trends. And I don't think, personally, it's just my opinion, that there's really strong evidence for that sort of relationship in terms of, you know, predisposing evidence. Am I answering your question correctly? Yeah. So that's my opinion. I don't know if anybody, Uta, do you want to comment on that?
No, I think it's nice talk.